I'm delighted to welcome first to the stage Giovanna Lombardi, who is the Professor of Human Transplant Immunology at KCL. And Giovanna is also the co-lead of our BRC's Advanced Therapies and Experimental Medicine cluster. And Giovanna is going to tell us a bit more about the early stages of advanced therapies development. So over to you, Giovanna.
Thank you very much, Kate. It's a pleasure to be here today. If I can have my first slide, please. Yes, I will present a long journey that started many years ago in the development pathway in cell therapy. If I can have the next slide, please. Now, I think we all agree that cellular therapy, in particular to manipulate the immune system, is rapidly evolving in the fight against cancer. You will hear more later on by Sophie. In the modulation of inflammation, and Ash will present something about this, and in the treatment of autoimmunity transplantation. And this is really my interest, and has been for many years. While in cancer, the immune system has to be stimulated, in autoimmune disease or in the prevention of transplant rejection, it has to be switched off. And we focus on a population of cells that I will tell you more in a moment, with regulatory function that has been very well characterized by us and others, and then exploited in preventing transplant rejection and treating autoimmune disease. If I can have the next slide. And these are the regulatory T cells, like all the other T cells of the immune system are thymus derived, are a very small percentage of the peripheral CD4 positive T cells, and they express some markers, some helpful for their purification, like CD25, and some that characterize them, like the FOXP3. Now, these cells are key in maintaining peripheral tolerance and preventing autoimmune diseases, and this has been demonstrated in animal models and in men, where the lack of this population of a defective function led to autoimmune disease. Next, please. Now, the idea that we develop together with many other people around the world is that if we can use these cells to tilt the immune system, we can reach a state of tolerance or regulation, and essentially is increasing the number of these cells that can compensate for the effector mechanism in many diseases like transplantation and autoimmunity. Next, please. The clinical vision that we developed more than 10 years ago, that if we take a blood where the cells are from end-stage organ failure patient that in need of transplantation, and then purify these cells using, for example, CD25, and expand the cells in a special laboratory, and then inject back in patients that receive either kidney or liver transplant, as I said, we can tilt the balance in favor of regulation. Next, please. This has been a very long journey and started, as I say, when the BRC started, and I have to acknowledge here the contribution and the financial help from the BRC. And we use blood from healthy control and also from patient. The example here is from a patient with liver cirrhosis. And essentially the Treg were purified using the CD25 by immunomagnetic beads. And here you can see the flow cytometry of freshly isolated cells, CD25, FOXP3. And then the cells were expanded for more than a month, polyclonal with anti-CD3, CD28, interleukin-2, and rapamycin. And rapamycin, we demonstrate already that was very important, as you can see in the right two panels, that the CD25 FOXP3 is very much upregulated when rapamycin is present. But most importantly, next please, is that rapamycin maintained the regulatory capacity of these cells from S1, that is the beginning, to the final stage. And these are all the close symbol. And this is independently in the suppression assay of the ratio between effector T cells and regulatory T cells. Next, please. Now, to demonstrate that these cells are really functional, we optimize a humanized mouse model where we finally tested these cells, human cells, in vivo. And if you start from the bottom, we use a immunodeficient mice, and these mice were reconstituted or transplanted with a human skin. And while the human skin was engrafted in these mice, we generated regulatory T cells with the protocol that I mentioned to you before, either polyclonal, as you can see on the right-hand side, but also we compare the function of these polyclonal Tregs with Tregs that were specific for the donor antigen by using antigen-presenting cells, either B cells or dendritic cells that express donor antigen. And then these regulatory T cells were injected together with effector T cells in these transplanted mice. Next, please. The human skin graft then was monitored, and we look for any sign of skin graft damage as a sign of transplant rejection. As you can see, this is one of the markers that we evaluate, Ki67, that is the proliferation of the keratinocytes that is characteristic when the skin is damaged. And you can see that there is a lot of green staining when PBMC or effector T cell alone are injected, but this is very much decreased when the PBMC were co-injected with regulatory T cells. And on the right-hand side, these are data from many animals and many analyses. And you can see again a lot of proliferation when effector cells alone are injected, and this is decreased by polyclonal and even more by donor-specific Tregs. And this is relevant, and I will explain more at the end. Next slide, please. Then this protocol for the expansion of regulatory T cells polyclonal was moved to the GMP facility, and I need to acknowledge a lot of people in the GMP facility have been key for the success of this program. And then the regulatory cells were purified, mostly using the CD25 expression with the Clinimax, expand with in bags for a month with the protocol that I mentioned before, and then the cells were frozen away until needed to be injected into patient. Next slide, please. And 2 clinical trials have been completed recently, and the outcome of these 2 clinical trials is that the Tregs are safe. These were one clinical trial with an EU consortium called the ONE study in kidney patient, and then the TRIL supported by the MRC-DPFS in liver. Altogether, we treated 21 patients, and as I say, we demonstrated the safety of this Treg. But not only, we had also some hints of efficacy, and in the next slide, I just show one example from the liver trial. And on the left-hand side, we demonstrated this again. I need to acknowledge the support of the flow cytometry platform. We managed to demonstrate that regulatory T cell can be visualized in the blood of this patient, in the sense that there was an increase after 2 weeks from the injection of these Tregs. But most importantly, on the right-hand side, is that we demonstrated that the donor-specific response was decreased after injection of these cells from baseline to month 1, while the response to third-party alloantigen or to recall antigen remain constant from baseline to month 1. Next slide, please. A few more clinical trials started since there are 2 clinical trials in transplantation.
Can I just ask a question? How can we have 3 out of 10?
Sorry, I can. The 2 studies, then game changer. These are in with transplant patient, in particular with kidney transplant patient. One in inflammatory bowel disease, Tribute, and Tiara, that is a clinical trial in aplastic anemia patient. All these trials use polyclonal Tregs with some minor difference in Tribute, and you can see in bold all the API of this clinical trial that I say just started. The next slide, please. I just want to finish by saying that obviously this journey hasn't stopped in the lab. We are still working very much on regulatory T cells, and going back to the concept that regulatory T cells specific for the transplant are superior compared to polyclonal Tregs, we came up and borrowed the idea from the cancer field that we generate regulatory T cells specific for the donor antigen using chimeric antigen receptor. This approach approach not only allowed to play with the specificity and have Tregs specific for any antigen that you select, but there are also additional advantages, namely that you can further manipulate these regulatory T cells to increase the phenotypic stability, the persistence, the safety, and also increase their function. And next, please. And all this work, as already Robin mentioned, led to a startup company, Quell Therapeutics, and I'm very pleased to say that by the end of this year, we will have the start of the first clinical trial with CAR-Tregs in liver transplant patient. Next and last slide, please. I mean, here are just some of the people that have been involved in this long journey, lab members, collaborators, and I have to mention a lot of more people in the BRC that have been support, like Mylene. And also this journey started, as I say, more than 20 years ago, and Robert Leckler has been present since the beginning. And thank you for listening.